Hello, this is Gina Piscatelli with a presentation on a lab topic that you're doing at home online, and that is eye physiology. In this lab, there's 4 main topics, and you can find these in the study guide on page 139. The 4 main topics include looking at the function of photoreceptors, which you already know a little bit about, rods and cones. We'll also look at the functions of the lens, the function of the pupil and the iris, and then finally we'll look at how information travels from both eyes to the visual cortex of the cerebrum, so all the way to the brain. This will involve some anatomy and some function. So let's start with the first learning objective. You have this picture on page 140. It's in the center of the page, and it shows a picture of both a rod and a cone. These are 2 different types of photoreceptors that have different shapes. They're of course located in the retina, and the rod you'll notice in the outer segment, which is pointed out here with this marking outer segment. The outer segment is very rectangular, whereas the outer segment of the cone has a point. That's how they get their names. Rods and cones are named by their shape. Now, both of these types of cells respond to light waves. They're stimulated by light waves, and in response, they will most likely send a signal to another cell. And that's very much like a neuron. So they're not neurons, but they're often referred to as neuroepithelial cells. Now, both of these types of photoreceptors contain visual pigments in this outer segment region. They're just proteins that are inside the cell. And these visual pigments change shape when they absorb light. So a light wave comes along and this pigment, visual pigment, changes its conformation or its shape. Think of how proteins can change their shape. Now, all of the rods that we possess in our eyes contain pigments that are stimulated by light waves that we think of as white light, but we see them in shades of gray. Okay, so dark versus light. That's what rods respond to. Cones, on the other hand, you've heard associated with color. There's actually 3 different types of cones, depending on what pigment they have. So some cones will have pigments that respond to blue wavelengths of light. Other cones have pigments that respond to green wavelengths of light. And then finally, there's a type of cone that responds to red wavelengths of light, or at least we perceive them as blue, green, and red. Now, a combination of those 3 types might be stimulated by any light source. So you might get a mixture. We don't see things just blue, just green, just red. Of course, we see purple, we see yellow, we see orange, and that just reflects the different cones that are stimulated by the light source. So when the pigment does absorb the light wave, it changes shape, and then ion channels open up. Ion channels embedded in the cell membrane open up. They might close, but we'll say they open up, and ions come in, and that triggers something to happen inside the cell. Usually, what we'll refer to in this lecture anyway, is that the cone or the rod will release a neurotransmitter. Okay, so once the visual pigment has been stimulated, this photoreceptor is activated to release neurotransmitter, or it's also possible that it's deactivated. But we'll just go with activation because it's easier to comprehend. Okay, so recall from our study of eye anatomy that the location where we have the highest density of cones is in the fovea centralis. That's directly behind the pupil. So if light waves fall here on the retina, we get the sharpest image possible and colored. Now, rods are located all over the eye except the fovea centralis. And there are also cones distributed in the entire retina as well, but the most concentration is in the fovea centralis. Now there's one region that doesn't have any photoreceptors, and that's called the optic disc. So if a light wave comes in and hits the optic disc, there are no photoreceptors to detect it. So you will not see whatever that light wave bounced off of. This creates a blind spot in our vision, in our field of view. Okay, one extra thing I want to add that is not in your study guide, but I think is really important, is that the layer, or the retina, has multiple layers. So this is the posterior side of the retina, so closest to the periphery of the eyeball. And these are epithelial cells that are dark, and that helps absorb light waves. And then we have the layer of photoreceptors. Then we have a layer of cells called bipolar cells. And then towards the anterior, or more superficial if you will, we have the ganglion cells. It's these ganglion cells that have axons that will travel to the optic nerve. So what's interesting about this is that when light comes into the front of the eye and hits the retina, it needs to travel through 1-2 layers of cells and then to the outer segment of the photoreceptor to cause any stimulation. It's very interesting how it has to go through those layers. But then the neural signal through the release of neurotransmitters and cell communication will travel in the opposite direction. So the rods and cones release a neurotransmitter that stimulates the bipolar cell or bipolar cells. And then the bipolar cells release a neurotransmitter that stimulates the ganglion cells. And the axons of these ganglion cells will form the optic nerve, which will travel to the posterior again. So I have another picture to show you that I think is a little bit more clear. So I like this picture. It's just so pretty. So let's say light comes in, passes the cornea, passes the lens, and hits the retina. Okay? When it hits the retina, it goes across the ganglion cells, the light wave goes across the bipolar cell, and then hits the photoreceptors. They are stimulated, and in response, they're going to send an electrical or neural signal in the opposite direction. So it'll go back anterior. And then you can see here the multiple axons that come from the ganglion cells travel to the optic disc and form the optic nerve. So this explains a little bit why the optic disc is not able to absorb light. It's because the optic disc is just full of axons. There's no photoreceptors there. So the take-home message is just that light must cross to the deepest layer of the retina to get all the way to the photoreceptors, and then the signal comes back around and goes out the optic nerve. If you'd like to look at a diagram that will help you understand this a little bit better, you can draw on a diagram you have on page 145. And this picture shows that light is coming from the bottom of the picture, so that's the light source. And you would just draw an arrow towards the photoreceptors. So now they're stimulated. Now the electrical signal has to get to the optic nerve. And that's why I've drawn in these purple arrows. The electrical signal will come forwards, travel kind of laterally to the optic disc and the optic nerve, and then go posterior. Okay, that's it for the retina. Now we're going to look at the function of the lens. The function of the lens primarily is refraction, and this is defined as bending light rays. And the purpose of bending light rays is that you've got light rays coming into the eye at all different sorts of angles. And so the lens will bend these light rays so that they converge, come together in the eye to produce a sharp image on the retina. We don't want it blurry, we want it to be focused. Now the lens is able to change its shape, which changes how much it bends light rays. This ability is referred to as lens accommodation. It still involves refraction, but it allows us to bend rays at an appropriate amount to see objects that are near as well as objects that are far, and bring them into focus. So let's just review what refraction means first. So we know that refraction is the bending of light rays. Why do light rays bend at all? Well, they're traveling at a certain speed in an environment. So if you look at this picture, which I wish you had in your lab manual, but you don't. But if you look at this picture, there's a light wave coming in the air, and all of a sudden it hits a new medium. It hits a new material. And it'll either slow down or speed up because the material will either be denser or not. If it's denser, it slows down. Anytime a light ray changes speed, it bends. So it hits this denser glass and it slows down and it's bent, it's refracted. When it leaves the glass material and goes back to the environment, it speeds up, so it's bent again. So the bottom line is refraction occurs as light rays enter and exit new materials. Now the cornea can bend light rays because, of course, the cornea is not the same material as the air that light waves are traveling through. So when light rays hit the cornea bends them because they'll slow down. But the cornea doesn't have the ability to change shape. So the rays or light waves will be bent at exactly the same angle all the time. No matter if the light wave came from a distant object or a near object, it's always, or what angle it comes in at, it doesn't matter. It'll always be bent the same amount. The lens, however, is what offers some flexibility. It can change the degree or the angle of refraction as needed for near objects versus distant objects. So now we're going to look at lens accommodation a little bit. So the purpose, as I mentioned, is to change the degree that light rays are refracted so that we can view near objects versus far objects. So to understand this, let's look at the picture on the right. The picture on the right shows you two lateral views of the lens. So I'll just put an L on the two lenses. Okay. Notice that the lens on the left is thinner. So a light ray coming across the cornea and then hitting the lens will be bent a certain amount when the lens is thin. But then look at the second picture over on the right. The lens is quite thick. That will bend light rays more. So the same light wave that comes in, crosses the cornea, hits the lens, but it travels through the lens a lot longer because it's thicker, and so it bends more. So a thicker lens will bend light rays more. And we use this ability, or our eyes use this ability, to see far objects when the lens is thin and see near objects when the lens is thick, or at least bring them into focus. You might be able to see a blurry image if the lens can't become thicker or thinner, but the ability for the lens to do this means we can bring both objects near and far into focus. Now, how does the lens change shape? It's all dependent on a muscle called the ciliary body or ciliary muscle. It's made of smooth muscle, so you can't control it. It's involuntary. And it surrounds the periphery of the lens. So this is a frontal view that I'm showing you here. And in a lateral view, you can see the ciliary muscle kind of up here underneath the iris in both cases, on both pictures. And you have this picture on page 141 if you want to look at that. So, on the bottom of page 141. So that's the ciliary muscle. That's where it's located. And when it is relaxed, it's kind of rolled backwards. And in this case, the diameter of the ciliary body or ciliary muscle is narrow. So it rolls backwards. Well, that pulls on suspensory ligaments that are attached to the lens. So the suspensory ligaments are shown as lines here in this picture, which you don't have this particular one, but you have the suspensory ligaments attached to the lens and the ciliary muscle in your figure on page 141. So the ciliary body has rolled backwards. It's relaxed. It's like reclining. It's happy. It pulls on the suspensory ligaments. They become tight, and that stretches or pulls on the lens. So the lens becomes thinner. This is what we use to view distant objects. Okay, so our ciliary muscle is in a relaxed state to view distant objects. But when we need to focus on objects that are near, the ciliary body or muscle contracts, and that means it rolls forward. It kind of widens. And you'll notice this frontal view shows you that the ciliary body or muscle is wider, thicker. That makes the suspensory ligaments slack or loose. And so the lens is not pulled as taut, or it's not stretched, and it balls up into its natural state. It becomes kind of rounded or globular, thicker. The lens becomes thicker. We use this for near object viewing, and we're going to talk about that next. So this figure is also on page 141 of your lab manual, just above the other one. And it's showing you how light rays are bent differently by a thick lens versus a thin lens. So in this instance, the object is exactly the same distance away from the eye. So we're not talking about near versus far here. We'll just talk about near. So the light rays bounce off of the object, which is a round red ball. They enter the eye, reach the lens, and they're bent. This is a thick lens, so they're bent quite a bit. And so light rays coming off the object from different spots are bent, and there will be a point of convergence where they come together in the vitreous humor, in the posterior segment of the eye, and then the light rays continue in the vitreous humor. Whoops, that's not a straight line, it should be, to hit the back of the eye, the retina. And this is where we receive that inverted image here. Okay, it's a ball, so you can't tell it's inverted, but that's where the image would land. Notice in the thin lens, the light rays come in at the same angle, but they're not bent nearly as much. And that's because the lens is thinner. You won't get as much refraction. And so the point of convergence is going to be much farther back, the back of the eye, and you won't get a full sharpened image at the retina if you're looking at something near. But the thin lens bends the light rays much less. So now let's put that concept together with how we view things that are distant versus near. So I'm going to take a lens that, let's see if you have this picture, sorry. Page 142, yes you do, at the bottom. Okay, so in this instance, I've got a thick lens. Okay, so the lens is thick. I'll put a T here, it's thick. And the eye is looking out at the field of view, and there are some objects that are distant, like this blue ball, and there are some objects that are near, like the red ball. Notice that a near object always looks bigger than a distant object. I think you know this intuitively. When something's close to you, it looks bigger than when it's far away. And so when an object is close and looks bigger, the rays come in farther apart than the rays from a distant object because it's smaller. So they need to be bent more so that they will converge. That's why the lens needs to be thick, because you're looking at a bigger object, so to speak, because it's close by. And so you need to bend the light rays more. And they'll converge, as I mentioned before, and you'll get a focused image on the back of the retina that's inverted. Okay, so rays are refracted more with a thick lens in order to see near objects. The image of the distant object, if you're sort of aware that there's other things out there than just what's right in front of your face, will appear blurry. And notice that the size of the retinal stimulation is much bigger. So you're going to produce a much blurrier image of that distant object. Okay, let's look at what happens when we try to focus on something that's far apart. In this case, we need a thin lens. Okay, and we need the lens to be thin because we want bending to happen less. We want the rays to be refracted less. This is because they're already coming in close together. So rays from a distant object come into the pupil or the eye closer together than something close to our eyes. So here are the rays coming in pretty close together. Since they're already pretty close together, they don't need to be bent quite as much to get this point of convergence and a nice, sharp, focused image on the retina. Notice what happens when you've got a thin lens and it can't change, let's say, and you're trying to focus on something that's close to you. The rays come in far apart, but they're not going to be bent very much They'll be bent the same way that the distance objects are, and they don't even converge within the eye because they haven't been bent enough. And the focused image will even happen out of the eyeball. Not that it ever gets out of the eyeball, but if it could, it would be focused beyond the length of the eyeball. So this just underlies the importance of needing to be able to change the shape of the lens to focus on either distant objects or far objects. Now there's a couple of visual impairments that are known pretty well, even in layman's terms, if not the actual medical terms. One is presbyopia. This is the type of vision that occurs with age, so the aging process. The lens loses its elasticity, and so it loses its ability to accommodate for near versus far vision or focusing. And so if you can't do that, the lens gets fixed within a much narrower range of how thick it can be or how thin it can be. And so people usually end up with bifocals. They use one part of the glasses to view something far away and another part of the glasses to view something distant. What's far more common, especially in younger people, but older people too, are nearsightedness and farsightedness. The correct medical term for nearsightedness is myopia. In this case, the person can see objects that are near, can focus on objects clearly better than distant objects. So distant objects don't focus on the retina because the rays don't converge properly in the right position to get a nice focused image on the retina. The cause is that the lens is too thick, or it could be that the eyeball is too long, the retina is too far away. So we add a corrective lens to the front of the eye, which is a concave lens. It's caved in on both sides, trying to draw, so that the rays that come in are spread apart before the thick lens bends the rays. So let's look at these lenses, and then we'll come back to hyperopia. So this is a picture of a concave lens on the right, and I'm sure you have this picture. You do. You have something like it on page 144. So this concave lens, notice the light rays come in and they're spread apart. And when they're spread apart, that enables a thick lens to go ahead and do the maximum bending that it already is set to do. Okay, they're spread apart. It's going to do maximum bending. Now, why does this help? It helps because the eye is set up for seeing objects that are close by. But what if you're trying to view something that's kind of distant? Well, the rays are going to be bent so much that the conversion point happens very far anteriorly. And so the image that falls on the retina will probably be too big and it'll be blurry. To correct for that, we put a concave lens in the front so that the rays come in farther apart. They come in farther apart. And that way the bending that occurs, that's pretty extreme, will push the point of convergence back farther. And so now the image will fall on the retina appropriately. Okay, let's go back to hyperopia and just look at the definition. Hyperopia is also known as farsightedness. So in the case of farsightedness, Objects aren't focused on the retina either, but that's because convergence happens too far posteriorly. In this case, the lens is thin or the eyeball is. So if the lens is thin, that works out very well for viewing something that's distant, but not for something that's near. So a corrective lens is used, called a convex lens, to be able to see an object that is closer. So here's the corrective lens for hyperopia. The condition, again, recall, is the eye is too short or the lens is too thin, and we're going with the lens option here. So the lens is too thin, perfect for seeing something distant, bad for seeing something close. So we're going to say this person is close by, and somebody's trying to look at it who's actually farsighted. The rays come in close together. Oh, I'm sorry, we're saying this person is near. Sorry. This person is near. The rays come in far apart. The lens cannot bend the rays enough. And so you get convergence too far back. In fact, the image usually lies beyond the eyeball. I mean, a focused image. You'll still get stimulation of the photoreceptors, but the image will look blurry, non-focused. So to fix this problem, a convex lens is put in front that helps the bending. So you have a near object coming in. This lens is too thin, the natural lens is too thin to bend enough. And so the extra lens helps. So you get some bending here, you get some bending here, and now we get a more anterior convergence, and the image formed on the retina is more focused. I hope I haven't confused you about nearsightedness and farsightedness. We'll do an exercise at the end to help you understand that a little bit better. Okay, so we're going to go to our third topic now, and the third topic relates to the function of the pupil and the iris. So you know that light rays have to enter the eye by going through the pupil, the round hole that appears dark, and the pupil is surrounded by the colored portion of our eye called the iris. The iris is made up of smooth muscle, and it controls the diameter of the pupil in order to change how many light rays enter the eye. Now, if you're in bright light, you typically limit the amount of light rays that enter the eye. That's the adjustment. If you're in dark environments, then you need to maximize how many light rays the eye can capture, and so the pupil will dilate. So let's look at how the iris can cause both constriction of the pupil and dilation of the pupil. This is kind of at rest, normal, the picture in the middle, which you have on page 143. And it consists, the iris here, notice, consists of two layers, a radial layer of smooth muscle and a circular layer of smooth muscle. Now, to constrict the pupil, it's the circular muscle that contracts. It's actually called a sphincter sometimes. So we call this layer the pupillary constrictor. To dilate the pupil, that circular muscle relaxes and the radial muscle contracts, and it pulls the iris kind of away from the pupil, making the pupil diameter larger. So that's how the iris works with its two layers. And the result for pupil dilation, and why we refer to this as accommodation again, is that when we're viewing near objects, a lot of light rays enter the eye because the object is so close. They come in at all different angles into the eye, and typically the pupil needs to constrict to just capture enough light rays to get a focused image. You don't want the entire retina to be stimulated. You'll have way too many neurons firing and the image will appear blurry. So in bright light situations, as well as focusing on near objects, the pupil constricts because the pupillary constrictor muscle, the circular muscle in the iris, has contracted. Instead, when we're viewing objects that are far apart, not as many light rays bounce off of that object and enter the eye. So the eye really needs to capture as many as it can to view distant objects. And this is true when you're in environments of dim light as well, or even in the dark. Your pupil will dilate to capture as many light rays as possible. This is achieved because the pupillary dilator layer, the radial layer of the iris, will contract and widen or dilate the pupil so that you can get as many light rays as possible into the eye. Now, this control of the iris is what we consider autonomic, meaning you cannot control it. It's controlled by your sympathetic nervous system and your parasympathetic nervous system, which we'll talk about in our next unit of lecture. So in summary, we looked at learning objectives 2 and 3, how the lens performs accommodation, and how the ciliary muscle or iris and pupil perform accommodation. So when you're trying to view distant objects, the lens accommodates by becoming thinner, and the iris or pupil accommodates by dilating. When you're trying to view something close, get a nice focused image of something near you, the lens accommodates by becoming thicker. That's due to the ciliary muscle, of course. And the pupil constricts, and that's due to the action of the iris. So you have an activity that we could do on page 151 of your lab manual. I'll give you a second to turn to that page. And you may wish to not write in there right now and just listen, or you can do the activity with me in either case. So the picture at the top shows how light rays are entering the eye that is in kind of a relaxed state, we'll say. Maybe the object's not super close, it's not very far. I don't know, it's the distance of your arm, maybe. You're holding it out. And the light rays come in. The lens is medium in thickness. We get nice convergence and a great focused image right on the back of the eyeball. In fact, this one's right on the fovea centralis, which is perfect. Okay. But how could we correct for a situation where the eyeball is too long? So with the same lens, the image, focused image, does not fall on the retina. It falls in the vitreous humor. Those rays will continue out and make a bigger image on the retina, and so it'll appear blurry. How can we fix that? Would we want to bend those light rays more, or would we want to spread them out? Well, we would want to spread them out. So typically what we would do is give someone eyeglasses that are manufactured with a concave surface. And so the light rays will spread apart a little bit. so that this lens, when it does its bending, because it bends quite a bit, okay, will cause the image to land on the retina. So that's the kind of lens we'd want to use, is a concave. Oh, yes, concave. Sorry. Oh, convex. I just said the word wrong. I'm sorry, you guys. Okay, convex. Getting myself mixed up with all the words. I'm really going to try to write this correctly. Hope you're taking notes while I'm pausing here. Takes a long time to write. I'm sorry. Concave. Because it's caved in. Oh, it's just not writing very well. Okay, so there's a concave surface, a curved surface right here. Okay, that's fine then. All right, now let's look at hyperopia. Sorry about that. Hyperopia, the eyeball is either too short or the lens is too thin, and so not much bending occurs. And when the light rays come in from a near object, there isn't very much bending, and the image doesn't even land on the retina. The focused image doesn't. There's still stimulation of the photoreceptors. It'll just appear blurry. So when someone is hyperopic or farsighted, we need to bend the light rays more, and so we use what's known as a convex lens. It doesn't like it when I write too fast. There, convex. Okay, that's one activity done. We only have one more learning objective to cover, and it has to do with the visual pathway. And by the way, these slides tell you the answers to those questions. And these slides are posted on Blackboard. Okay, so the last learning objective has to do with the visual pathway. That means it refers to the neural impulse that comes from the eye and goes to the brain so that we can actually perceive what it is our eye is seeing. So there's different components of the visual pathway. You're already familiar with the retina, and you're familiar with the optic nerve coming out the back of the eye. And because you've studied the brain already, you're familiar with the optic chiasm, where the two optic nerves meet. And then from that point backwards, we haven't talked about much. So from that point posteriorly, we have the optic tract, and then neurons kind of spread out, so we call these optic radiations because there's pathways of multiple neurons spread out. And then you reach finally the cerebral cortex in the occipital lobe. So we call it the occipital cortex or the visual cortex, either one. So those are the anatomical components of the visual pathway. And the reason we're interested in studying this is because we perceive our field of view, what we're looking at, the environment we're looking at, that's our field of view, on opposite sides of the brain. So if this person has an entire field of view in front of them, both eyes are able to capture some information from the left side and some information from the right side. Both eyes get light rays from both sides. But where is that information sent? It turns out that all information from the right field of view goes over to the left side of the brain from both eyes. And I'll change color here. From the left field of view, information is going to go to the right side of the brain from both eyeballs. Okay, so we're going to study how that can be possible. It's anatomical mainly. Okay, so this picture is shown on page 145, and it has, I've changed it a little bit by placing a box with a field of view in front of it or on top of it where it says the visual pathway in your lab manual, and color-coded the field of view. So left versus right. Okay, so you can see already how it's set up. The green field of view goes to the opposite side of the brain, and the orange or salmon colored field of view goes to, again, the contralateral side of the brain. And that has to do with the pathway of neurons from the retina through the optic nerve and through the optic tract. So first I want to mention that optic nerves coming from both eyeballs carry information or signals from both sides of the field of view, right and left. And if you look closely, you will see that there are some salmon-colored axons in there, and there's some green-colored axons in there. Same for this right eye. Okay, both colors are in the optic nerves. But now look at the optic tracts. These optic tracts only have neurons carrying information from one side of the field of view. This optic tract has only information from the right side of the field of view, and this optic tract only has information from the left side of the field of view. So it gets split up in the optic chiasm. That's how it happens. And we're going to trace that pathway so that it's easy for you to predict what would happen to vision of the field of view if you had damage to an optic nerve versus an optic tract. It would be different. You'd have different results, different problems. Okay, this picture is found on page 146, and it's the beginning of how I'd like to teach you to understand these different pathways and how they travel. So again, we have a left eye and a right eye viewing both the left and right fields of view. So this is the field of view. This is what you're looking at. And I have labeled, and you might want to do this too, I have labeled two sides of each eye, left and right, with the white letters L and R at the back of the retina there. So you might want to do that. Okay, so the field of view is labeled left and right. The eyeball retina is labeled left and right. And of course, overall, this is the left eye. And on the other side, because this is a superior view, this is the right eye. Stupid thing. Right eye. Okay. All right. The way that you want to attack this to understand how the information is moving, and I'm going to use yellow so that you can see what I'm doing, is you want to draw from the left field of view light rays entering both eyes. So notice that they go in at an angle, and it's the same angle into both eyes. So the right retina of both eyes is stimulated by the left field of view. Now I'll use orange from the right field of view. And you'll notice that, again, light rays from the right field of view come into both eyes at an angle, hitting the left retina of both eyes. So on the left eye, the left retina is stimulated by the right field of view, and in the right eye, the left retina is also stimulated by the right field of view. Okay, so now we just trace the pathway through the optic nerve using the appropriate colors because it makes it easier. So I'm just going to start with orange. Since we're over here in the lateral eyeball, it stays, that information, that signal stays in the lateral part of the optic tract, and it bypasses the optic chiasm and goes directly to the brain. But that's not true for the signals being carried from the medial part of an eye. In the medial part of the eye, the electrical signal is traveling in the medial part of the optic nerve, and it reaches the optic chiasm and enters it, and crosses over to the opposite side. So let's do the other eye as well. Let's start with the lateral right side retina of the right eye, and notice that the signal from the left field of view and the right retina stay pretty lateral, bypassing the optic chiasm and going in to the right visual cortex. Whereas information that is traveling from the medial portion of the right eye stays in the medial optic nerve, hits the optic chiasm, and crosses over. So, since orange came from the right field of view and yellow came from the left field of view, or green and pink, right? Depends on what colors you're using here. Information from the right field of view, both eyes have sent that to the left visual cortex. Whereas visual information sent through both eyes from the left field of view goes to the right visual cortex. So the last thing to do is there's an exercise in your lab manual where you can practice this. And so you want to be able, what the objective is you want to be able to answer a question something like, I'll type one out here. Vision from which field of view would be impaired when the left optic tract is severed. That's terrible. You don't want that to happen to anyone. But let's just see. I'll make a cut, a random cut. Now, to do this activity, you're probably going to want some colored markers. Blue and yellow are ideal, but whatever. So I'm going to cut the left optic tract here. Cut. Nothing's getting past this. Okay, so that's, I don't know if there's a tumor or there was a traumatic brain injury or what, but regardless, this has been cut. Okay, so right away, I think you already know that it's going to be the right field of view that we would not be able to perceive, right? Even though both eyes are working, you can't see the right field of view without turning your head and making whatever was in the right now on your left side. That's the only way that the brain would make it work. So I recommend that you work on this, practicing drawing the pathway. I do have an answer key for you posted on the next slide, and we can do it during our live lab session as well. Okay, it's just getting kind of long. I'm pretty sure you don't want to listen to me anymore. So please practice drawing those light rays in this diagram. Any other activity that you see in your lab manual, like determining your near point, the visual acuity, testing for astigmatism, test for color blindness, all that stuff, we aren't going to cover. You're welcome to read it and work on it with anybody that you're friends with, but it won't be covered on the test. Thank you very much.